Welcome back, biologists. It's Mr. Forberg here, and today we're looking at a long free response question coming from last year's 2019 exam. What I see right in front of me is a real quick enzymatic pathway followed by some text, so let's get started. I've already scanned through parts A through E, and I can tell that parts A through C are directly related to the diagram itself, where D and E are going to propose a new scenario and ask me to kind of think about what's going on. So let's look at the text itself to see how it supports this particular enzyme pathway. It says that auxins are plant hormones that coordinate several aspects of root growth and development. Indole-3 acetic acid, or IAA, is an auxin that is usually synthesized from the amino acid tryptophan from figure 1. The gene TRPT encodes an enzyme that converts tryptophan to indole-3 pyruvic acid, or I3PA, which is then converted to IAA, an enzyme encoded by the gene YUC. We can see all the components that are sitting right in the pathway above. We're going to start off with tryptophan right over here on the left. We know that it requires an enzyme to be converted to I3PA, and then a second enzyme YUC to get to our final IAA, which again is a hormone that's going to promote root growth in plants. We know that these enzymes are coded for by genes, and we can see that we've got genes for each enzyme, right? And then we're producing an mRNA transcript, which will later be translated. Let's take a look at the very first task verb. In part A, we're being asked to circle one arrow that represents transcription on the template pathway, and then identify the molecule that would be absent if YUC is non-functional. The very first part says circle, and I really want to point out where it says one arrow. Anytime they direct the number of items they're looking for, don't exceed it. They're going to look for the first one that you put down. If one contradicts the other, they can't give you the credit for it. So if they say circle one, circle one. Very simply put, I'm going to go ahead and see that here's my gene. So this arrow right here represents the transcription of that gene to produce the mRNA. Now, I could have also selected this arrow over here, but I'm going to dot that in there because if this was my response, it said circle one, I'm only circling one. The second part of this is I need to identify the molecule that would be present, or I should say that would be absent if YUC is non-functional. So we want to know the molecule under the condition of YUC being non-functional. So taking a look at the pathway itself, we can see that here is our enzyme YUC. If we eliminated this from the pathway, we know that we cannot convert I3PA over to IAA, and so this guy right here will not be present. So our molecule that's going to be absent is IAA. Let's move on to part B. Part B states, predict how a deletion in one base pair in the fourth codon of the coding region of the gene TRPT would most likely affect the production of IAA. Justify your prediction. The next piece that we really want to focus on is this prediction. So we're focusing on the prediction of a deletion on one base pair in the fourth codon. And here's the pieces that I'm really focusing in on. It's the deletion of a single base pair in a codon very early on in an amino acid sequence. Most most proteins are constructed of hundreds or thousands of amino acids, which means that if you have an error in a very early codon, you're most likely going to see that error continue through all the remaining codons. On top of that, it's specifying that the deletion is only one base pair, and one or two base pair deletions or additions are notorious for producing missense mutations that cause a change in the reading frame, which tend to be disastrous for the protein themselves. So if I want to predict what a deletion in a base pair in a fourth codon will have on the TRPT gene on IA, I'm going to assume that it's probably going to reduce or produce no IAA. So very simply put, my prediction, no IAA produced. Justify why? Well, if this particular mutation is affecting this mRNA transcript, I'm likely to see very little functional TRPT enzyme. Therefore, if I don't have the TRPT enzyme, I can't generate the I3PA necessary that's a precursor for the production of IAA, and there's my justification. We're ready to move on to Part C. Part C is going to focus on explaining one feedback mechanism by which a cell could prevent the production of too much IAA without limiting IP3 production. Simply put, my task verb explain a feedback mechanism that's going to allow us to reduce IAA or limit its production without affecting, again, I3PA. So the first thing that I do is, what part of the diagram do I want to focus on? Well, I don't want to mess around with this right here, so I'm going to focus on everything in this region of the diagram. I'm going to ignore everything to the left because I'm trying not to affect I3PA. Therefore, typically when we have a regulatory mechanism in place where we see that a molecule like IAA is limited in its production, it's because of negative feedback. So I would first identify that the feedback mechanism is going to be a negative feedback mechanism, and it's that the product itself, IAA, could be doing a couple different things. It could be negatively regulating the enzyme activity of YUC. So IAA could be a competitive or an allosteric inhibitor of enzyme YUC. Also, we could see that there could be some inhibition or regulation of the expression of the YUC gene. So maybe the production of IAA is going to repress the expression of the YUC gene, therefore we won't get any enzyme YUC. Both of those are acceptable answers. Now that we've got A through C done, let's take a look at D and E. Scrolling down, Part D is going to indicate that rhizobacteria are a group of bacteria that live in nodules on plant roots. Rhizobacteria can produce IAA and convert atmospheric nitrogen into forms that can be used by plants. Plants release carbon-containing molecules into the nodules. Based on the information, identify the most likely ecological relationship between the plants and the rhizobacteria. Describe one advantage to the bacteria of producing IAA. So when I think about what is a significant exchange that's taking place here, I note that the rhizobacteria themselves can convert atmospheric nitrogen into forms that can be used by the plants. That seems like it's a really good benefit for the plant. And likewise, the plant is going to release carbon-containing molecules into the nodules. And if I'm a bacteria, I need those carbon-containing molecules to survive. Much like you would eat a hamburger to go ahead and sustain yourself, that's how the bacteria are going to sustain themselves. So it sounds like this is a win-win scenario. So let's take a look at the task verb and see what they have us do with this. We're going to first identify, let me change color on that. We're going to identify the most likely ecological relationship between the plants and the rhizobacteria. Very easily put, if both organisms are going to benefit, we know that this is going to represent mutualism. The second task that we've got to do for Part D is to describe one advantage to the bacteria of producing the IAA. Well, very simply put, the more IA they produce, well, we know that IAA is going to stimulate root growth. So they're going to increase habitat, or you could say it could increase access to resources. More specifically, I might add that if the plant roots are growing, they're getting access to more resources themselves. The better the plant grows, the more carbon they can send to those roots and those nodules, which would be a benefit for those bacteria. Let's move into the very last part, Part E, and see what that has to say. Our researcher removed a plant nodule and identified several cheater rhizobacteria that do not produce IAA or fixed nitrogen. Describe the evolutionary advantage of being a bacterial cheater in a population composed predominantly of non-cheaters. Plants can adjust the amount of carbon-containing molecules released into nodules in response to the amount of nitrogen fixed in the nodule. Predict the change in the bacterial population that would cause a plant to reduce the amount of carbon-containing molecules provided to the nodule. The first task verb that I see is I want to describe an evolutionary advantage of being a bacterial cheater. Now, one of the very most important pieces of this that I really pay attention to is anytime they say evolutionary advantage, we want to make sure that we connect it to reproductive success. So not just an advantage, because an advantage would be, well, they get access to carbon-containing molecules so they can grow, and that's great, but how is it an evolutionary benefit? And so to make it an evolutionary benefit or advantage, we'd have to extend that thinking to, well, they don't have to expend resources to produce IAA for the plant or fix nitrogen, but they do get access to the carbon-containing resources to grow and reproduce at a greater rate. And that'll get you that point for that description in part E. Moving on, the second piece, we want to predict the change in the bacterial population that would cause a plant to reduce the amount of carbon that we're sending to the nodule. So if I'm a plant, why am I going to stop sending carbon molecules to a particular piece of the root or a nodule? It's because it's not fixing nitrogen. So what's the population that's actually fixing the nitrogen itself? Well, we know it's those non-cheaters. So if the non-cheater population is decreasing, we're going to see a reduction in the amount of carbon-containing molecules sent to that particular part of the plant. Or likewise, you could probably infer that a relative increase in cheater bacteria would lead to relatively less nitrogen fixation and IA produce, therefore, less carbon moving to those particular nodules. And that takes care of parts A, B, C, D, and E of the 2019 first free response question. Thanks for tuning in, and we'll see you next time.